Hello, everyone. I am Dr. Muzamil Liaqat Ali, Assistant for Biochemistry. Welcome to lecture number 3 in our Carbohydrate Metabolism series. In lecture number 2, we studied the complete pathway of glycolysis, its 4 stages, energy yield, aerobic versus anaerobic metabolism, and the clinically important Cori cycle. Today, we move to the regulation of glycolysis, the fine-tuning mechanisms that control the rate of this pathway according to the energy needs of the cell and the hormonal status of the body. A regulation of glycolysis is of prime biomedical importance because it determines how efficiently glucose is used for energy production, and its derangements underlie hemolytic anemias, lactic acidosis, cancer metabolism, and metabolic responses to exercise and shock. So let's examine each regulatory step with clarity, precision, and strong clinical correlation. As you can see on your screen, this illustration shows the glycolytic pathway. Let's talk about the biomedical importance of glycolysis. Glycolysis is the major pathway for glucose metabolism in the human body. It occurs exclusively in the cytosol of virtually all cells and possesses a unique feature. It can function both aerobically and anaerobically, depending on oxygen availability and the integrity of the electron transport chain. RBCs, the red blood cells, which lack mitochondria, rely completely on anaerobic glycolysis for ATP production. This pathway also serves as the principal route for the metabolism of fructose, galactose, and other dietary carbohydrates. So, what about the clinical correlation at this very point? In clinical or in skeletal muscles, during intense exercise or in hypoxic conditions, glycolysis becomes the only rapid source of ATP, allowing continued contractions even when oxygen supply is limited. Glycolysis is especially critical because it provides ATP anaerobically. In cancerous cells, glycolysis proceeds at a dramatically accelerated rate, even in the presence of oxygen. That is the Warburg effect, producing a large amount of lactate. This creates an acidic microenvironment that promotes tumor invasion and metastasis and has direct implications for cancer therapy. That is one of the targeting glycolytic enzymes, right? So, deficiency of any glycolytic enzyme most commonly manifest as hemolytic anemias. RBCs cannot generate sufficient ATPs in that condition, that is hemolytic anemia. And when the defect is in the skeletal muscles, patients presents with exercise-induced fatigue and cramps. So, what about the regulatory enzyme for the glycolysis? There are 3 in number, right? So, the flux through the glycolysis is controlled at 3 irreversible, that those are rate limiting steps catalyzed by hexokinase or the glucokinase. The second one is the phosphofructokinase 1, the most important control point. And the third one is the pyruvate kinase. So these enzymes are regulated by allosteric effects, substrate availability, and hormonal signals. Mastering these regulations is essential for understanding metabolic flexibility in fed versus fasting states. So these are the 3 regulatory enzymes of the glycolysis, right? Those are considered as the rate limiting steps and are irreversible. So let's talk about the phosphorylation of the glucose by the hexokinase versus the glucokinase. So this pretty slide compares the 2 enzymes that phosphorylate glucose to glucose 6-phosphate. Let's talk about the hexokinase first, present in most tissues. So, what are the features of hexokinase? Broad substrate specificity, low Km value means high affinity for glucose, low Vmax, and strongly inhibited by its product, glucose 6-phosphate. That is one of the feedback inhibition process, right? And what about the glucokinase? That is present in liver and beta cells of pancreas. What are the features? Those are highly specific for glucose, high Km value means low affinity, means functions only at high blood glucose levels, high Vmax value, and acts as a glucose sensor in the pancreas and allows the liver to phosphorylate glucose efficiently after meals. So, glucokinase mutations cause MODY2, that is mild familial diabetes. Hexokinase deficiency is a rare cause of non-spherocytic hemolytic anemia. So what about the regulation of the glucokinase by fructose 6-phosphate and glucose molecule? In hepatocytes, glucokinase is regulated by glucokinase regulatory protein, that is GKRP, glucokinase regulatory protein located in the nucleus. In the presence of fructose 6-phosphate, F6P, glucokinase bind tightly to the glucokinase regulatory protein and is sequestrated in the nucleus, and that leads to enzyme is inactive in this condition. When blood glucose rises, glucose enters the nucleus, displaces the fructose 6-phosphate and releases glucokinase into the cytosol where it becomes active. This mechanism allows the liver to rapidly respond to the postprandial hyperglycemia. What about the condition in which the blood glucose level rises, right? So let's talk about the regulation of glucokinase by insulin. Glucokinase is also under hormonal control, as we know earlier, right? Increased blood glucose levels means there will be increased insulin secretion. It means that undergoes increased transcription of the glucokinase gene, and that leads to higher enzyme levels. This long-term regulation ensures that the liver efficiently stores glucose as glycogen and fats in the well-fed state, right? Now, what about the phosphofructokinase 1, the committed step of the glycolysis? Phosphofructokinase 1 catalyzes the irreversible phosphorylation of the fructose 6-phosphate to fructose bisphosphate molecule. It is the rate limiting and the most important control point of the glycolysis. What about the allosteric regulation of energy status? Allosteric regulation by energy status. It is done by 3 ways, right? The very first one is it is inhibited by high ATP levels. Means that signals energy sufficiency. And the second one is it is activated by high AMP levels. Means it signals energy deficit. And the third one is it is also activated by fructose 2,6 bisphosphate and fructose 1,6 bisphosphate. That is feed forward activation process. Right? So phosphofructokinase deficiency, that occurs in Torres disease and one of the glycogen storage disease type 7, causes severe exercise intolerance and hemolytic anemia. What about the regulation of pyruvate kinase? Pyruvate kinase catalyzes the final step. That is the phosphoenol pyruvate to pyruvate formation, generating ATP molecule. Its deficiency is the second most common cause of hereditary hemolytic anemias. Regulated by CAMP-dependent protein kinase that is activated by the glucagon, and the phosphorylation inactivates the enzyme. So high glucagon levels that occurs in the fasting state therefore inhibit the glycolysis and favor gluconeogenesis, that is the formation of new glucose from the non-carbohydrate sources like the protein and the fats. And in liver, this regulation prevents futile cycling between glycolysis and gluconeogenesis. Now let's talk about the reduction of pyruvate to lactate. In tissues with the limited oxygen, that is in retina, lens, cornea, kidney, medulla, testes, leukocytes, red blood cells, skin, and GIT, or during intense exercise, pyruvate is reduced to lactate by lactate dehydrogenase, that is LDH enzyme. This regenerates NAD+, allowing glycolysis to continue anaerobically. So, exercise or in excessive lactate production leads to lactic acidosis, a life-threatening condition frequently seen in circulatory collapse, leads to circulatory collapse and shock. This severity of shock can be assessed by measuring serum lactate levels. So, what about the hormone regulation of glycolysis? The very first one is the allosteric regulation by the ATP, AMP, or some of the other molecule like the G6P molecule, right, is rapidly and it's short term. The second one is the hormonal regulation that is slower but more profound. Hormone regulation includes the insulin. Insulin promotes glycolysis in well-fed state, and the counterpart of the insulin is the glucagon and the epinephrine that inhibits the glycolysis and stimulate the gluconeogenesis that occurs in the fasting or stress state or in low energy states, right? This slide, the fate of the pyruvate, that is one of the major metabolic fates of the glycolysis that can be asked in exam frequently, and it's of high yield exam topic. What about the fates of the pyruvate? Pyruvate has four major metabolic fates. The very first one is the oxidative decarboxylation that occurs as the acetyl-CoA molecule in the presence of oxygen enters the TCA cycle. The second one is the reduction, the lactate formation that occurs in the anaerobic conditions. The third one is the amination that leads to alanine formation, the amino acid formation. The fourth one is the carboxylation that leads to oxaloacetate formation that occurs in the gluconeogenesis, right? This slide beautifully summarizes how pyruvate links glycolysis to other major metabolic pathways. So thank you very much for watching this video. We have now completed a comprehensive study of the regulation of the glycolysis from the three key enzymes and their allosteric hormonal controls to the biomedical importance and clinical consequences of the dysregulation. You now understand why glycolysis is tightly regulated, how it adapts to oxygen availability and hormone signals, and why deficits in this pathway cause hemolytic anemias, lactic acidosis, and contribute to the Warburg effect in cancers. In the next lecture, we will explore the fate of the pyruvate in great detail and enter the citric acid cycle, that is tricarboxylic acid cycle. Thank you very much for your attention and for watching this lecture. If this session clarified the regulation of glycolysis and its clinical relevance, please like the video, share it with your fellow medical students and colleagues, and subscribe to the channel for the complete high-yield medical biochemistry series. Turn on the notification bell so you never miss the next lecture. And further, drop your questions and doubts and interesting clinical cases in the comment section. I read and respond to every single comment personally. Which regulatory enzyme and clinical aspect would you like me to explain in more detail? Please let me know. This is Dr. Muzamil Liaqat Ali signing off. Revise these regulatory enzymes, their allosteric effectors, and the differences between hexokinase and glucokinase daily. Stay disciplined, stay precise, and keep mastering carbohydrate metabolism with passion and excellence. I look forward to seeing you in the next lecture. Thank you very much.